This method can help answer key questions related to the biophysics of mammalian ion channels, including, but not limited to, the Bestrophin family of ion channels.
The main advantage of this technique is that it can be applied to other types of ion channels and generates key products for various downstream in vitro assays. The implications of this technique extend toward therapy of bestrophinopathies because assays using the purified protein contribute to our knowledge of how bestrophin channels work in the retina and how mutations in the BEST1 gene cause macular degeneration.
24 hours before viral infection, add an equal volume of trypam blue to a 15 microliter aliquot of HEC293F cell culture and check the cell density and viability using a hemocytometer under a microscope. After performing the cell count, plate 0.6 times 10 to the 6 cells per milliliter in a 500 milliliter volume in each of 2 disposable 2-liter flasks. Place the flasks into a 37 degrees Celsius humidified incubator with 8% carbon dioxide. Rotate the culture on an orbital platform at 135 rpm. On the day of infection, check the cell density and viability of a 15 microliter aliquot of the culture as before. A high cell viability of over 95% is essential for efficient infection and protein expression. The expected cell density is about 1 times 10 to the 6 cells per milliliter, and the expected total number of cells is about 1 times 10 to the 9. Add the P3 baculovirus to the cells at a multiplicity of infection of 5. To determine the amount of virus to inoculate, use the equation shown on screen.